Hey, good morning, everyone. Today's guest and lecturer is Professor James DiCarlo. Jim is a professor at the MIT Brain and Cognitive Science Department, and in fact, until just a few weeks ago, he was the head of the department, I believe, for almost 10 years. Now Jim is the director of the MIT Quest for Intelligence. Jim, welcome to the BMM summer course.
Thank you, Boris. I hope you're enjoying the course so far. So this isn't meant to be a lecture that's as interactive as can be. I don't want to just spit at you here remotely. So I'll try to pause and then we'll take questions as often as I can. But please put up your hand and someone will find you with the microphone. And somebody please let me know if you stop hearing me broadcast. So I'm going to try to give you a talk I titled, I noticed in the presentation in the symposium listing, I was listed as primate mechanisms of object recognition. And that's fundamentally what my lab works on, but I view that as part of a larger quest to create engineering quality models of the mechanisms of human visual recognition. And a big part of my talk is trying to give you a sense of a kind of a philosophical approach of how engineering is helping neuroscience to achieve its scientific goals. And I hope that comes across in this example of vision. And there's a broader sense here that I think connects to the CBMM mission and many of your interests, I hope, as you get into this intersection between neuroscience, cognitive science, and artificial intelligence. So again, that's the title of my talk. I used to kind of title my talk Reverse Engineering, or a core component of human visual intelligence, but I want to be clear that when I say reverse engineering, it means essentially this. The goal is to create engineered quality models in the service of science. So let me step back and ask kind of all of you a bit, or just sort of frame the setting here. Why, this is CBM summer school, like why are you here? Or more broadly, not just the summer school why you're here, but what astonishes you? What questions drive you to want to even be interested in the mind and the brain? Now, I'm sure you all have your own thoughts about this. I want to provoke you a bit. And here, what I'm going to do is I'm going to show you a video. As Boris mentioned, I've been the department head. I just stepped down to take on the Quest leadership, but I was department head for almost 10 years, a little over 9 years. The Department of Brain and Cognitive Sciences, which used to be the Department of Psychology. And that was founded by a gentleman named Hans Lucas Teuber. And I'm going to show you a video of Teuber teaching his intro undergraduate course about psychology. And I'll show you that here. It's probably fun to see that, the sort of founding of our department in our field, but also motivating a question for you. So hopefully this audio will work. It's just a 2-minute long video if you can watch. Okay, here you go. So here's Teuber in 1974. It's a little grainy.
In a way, we could say that the first step in any new field is to know what to be astonished at, what to wonder at. Theoretical physicist by name of Murray Gell-Mann, who was here on a visit from that strange place on the West Coast, Caltech, our competitors. We tried very hard to persuade him not to go back there, but we failed. Gell-Mann said to me, without any provocation on my part, that he thought ours was a weak-kneed department. Rather than being unorthodox, as I claim, because it had in it neuroanatomy and neurophysiology and linguistics and other things that are usually left out, it was, not bold enough, because we were not studying those issues in psychology that were really important. I said, which ones are important? And he said, and he absolutely floored me by saying that, because I think he's bright. I know he's bright. He said, well, you shouldn't study perception, you should study extrasensory perception. You should not study language, but thought transference and hypnosis. And there was one third thing, which I have forgotten or repressed, as some people say. It might come back under hypnosis, maybe. But in any case, I was astounded and shocked because he said all this in front of our then provost. Fortunately, a very good friend of mine. I tried to convey to him the idea that the thing to wonder about is perception. That if sensory perception did exist, I would feel somewhat let down. I'm not against its existing, but I would feel deflated because it would be much easier to understand. Perception is a great river. How do I see a line as a line? And a face as a face? And as that face amongst 1,000 faces, where emotion and memory enter, that's a much greater river.
I hope that was fun for you to see. Teuber, as department head, really had a vision of setting up some of these questions that, not these sort of like, let's call them sort of UFO style questions of psychology at the time, but really trying to get at the mechanisms of things that we take for granted, perception being one that he highlighted here. And that's a big core part of the talk I want to give you today, is both to motivate you why this is an interesting problem, but then we've gone beyond what Teuber was able to do at that time. He could motivate the question, but now we've actually made progress on understanding those mechanisms. And there's still a long way to go, but we're on that road that he painted nearly sort of 40, 50 years ago when he set up the founding of the department. And so let me just start by reminding you, visual intelligence, what he's calling perception, which we now maybe call broadly visual intelligence, is really an astonishing feat. If you look at this image right here, you might say, well, you know, you can obviously tell me that there are cars or people or so forth. But when you get right down to it, these are just little pixels of light that are of different values. And that's what a computer sees when they see this, is just a whole array of millions of pixels that are out there in the world. And all the rest of this is a creation of our brains to give rise to our inferences that there are things out there in the world. Things like people and cars and so forth. So we can answer questions like, what's in this scene? Where are the cars? Even knowing there are cars to begin with. Where are there people? Again, knowing that there are people at all, and where they are and their poses and so forth. These are things that we infer very quickly. Beyond that, other questions of what will happen next? And that's related to, where is it safe to walk? And this is, of course, why this is an important ability that we have. And I think this was highlighted in both Co and probably Tiago's talks that they've already given you, of these kind of broader questions of intelligence. There's many other questions you can ask from this, but I want to motivate that this is not a trivial thing to do. And Teuber saw that. So that was quite astonishing to him as a goal, a scientific goal, just understanding perception. And I think the other thing that's astonishing, I think, to many people still today, is that this device here that you see now removed from its casing, this is a human brain, is really the seat of enabling that. So this is a quote, this is actually called The Astonishing Hypothesis from Francis Crick, and I'll read it to you. He wrote, "You, your joys, your sorrows, your memories, your ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules." Now, many people today still find this quote quite jarring and astonishing, because they think of the mind as something that's separated from the mechanisms of the brain. That's why they find it astonishing. But neuroscientists and cognitive scientists take it for granted that this, of course, is true. It's sort of a founding hypothesis of our field, that somehow those mechanisms of neurons and neural networks and synapses are what enables things that are astonishing, like perception and emotion and free will and all of that. So I just want to give you that perspective, but I want to also point out the limitations of this kind of quote. Even though it motivates things, again, as Teuber motivated things, I'm going to give you a similar quote for a device that probably most of us have in our pockets right now. So this quote here, you're looking at your iPhone, "Your remarkable favorite app, all its performance and amazing user interface are in fact no more than the behavior of a vast assembly of transistors." Now, of course, this is a true quote, but it's inadequate, right? So you can say that of course this is true, but it's not helpful in an engineering sense to say know how to intervene, to know how to predict what's going to happen next inside either of these machines. To do that, you need much more sophisticated models. So these are motivating hypotheses, but they're not really scientific hypotheses in the sense that they don't offer strong predictions, other than if you throw out the brain or you throw out the phone, the app won't work anymore. But getting into the details of the mechanisms is what the field is all about. And how do we model that? So I hope this is motivating for all of you. And again, partly why you're all here. So I'm saying all this as kind of a philosophical statement to kind of remind you of what kind of scientific answers our field is even seeking as we try to, quote, understand the brain and how it gives rise to the mind. So again, the phenomenon of human cognition, whether it be visual intelligence, as I've been motivating from Teuber here, or navigation, memory, social cognition, whatever phenomena of human minds that you're interested in, They are all emergent phenomena of some specific set of mechanisms, that is some specific neural network and the body that it's encased in that give rise to that. Again, this is the founding hypothesis of neuroscience. All of those emergent phenomena can and will someday be understood in the same way the phenomena of any engineered complex machine are understood. And this is an important point here. So they won't be understood in simple one-lines or abstracts or one great paper. They'll be understood as reproducible, engineered quality specifications of how the component parts interact to produce the emergent phenomena of interest. That is the behavior of interest, say, for instance, visual intelligence. Okay, I hope, again, most of you take this as kind of dogma, but I want to point out that's not a kind of all 100% standard view, even in the field of neuroscience, that we need to build those kind of systems. So to give you sort of that analogy, again, the transistors give rise to the amazing user interface, but there are really system level understanding to go from transistors to how that user interface works that we humans kind of maintain as global knowledge when we construct, improve, build these devices. And similarly, going all the way from neurons and synapses up to behavior and cognition, there are many levels here that need to be integrated. So we need multi-scale models that can go from neurons to synapses all the way up to behavior. I don't mean to imply that there's a one-to-one correspondence between what's at the top and what's at the bottom, other than to say that there are elements that we know a lot about, whether they're transistors or whether they're neurons and synapses, but that the collections of those elements are what's important to giving rise to these emergent phenomena. And that's really what's exciting about the field now, is that it's building systems levels models to engage on some of those phenomena. And today I'll tell you about, I'll give you a review of what you've already heard about some phenomena in visual intelligence. So I call these systems level model at multiple scales. Okay, so then this sets up what I called at the beginning, the goal of reverse engineering, as I called it, is the goal is to ultimately account for every ability of the mind, which we'll call intelligent behavior, say account for visual perception or even just object recognition, using a system of components of the brain, which would be systems of connected neurons as hypotheses, and to do that in the language of engineering, meaning we don't just say there's a collection of neurons that give rise to it, but we actually build such systems so that they can offer predictions and explain what's going on and tell us how we can intervene in that system in a predictive way. So often this is the way I think about this is brain and cognitive scientists are wanting to account for these things. That's what astonishes us. That's on the left here. Engineers are used to trying to build such systems. And what's exciting right now is that these fields are coming together, science and engineering, around building collections of connected neurons or neuron approximations that then are hypotheses of how aspects of intelligence may work. And I refer to those as intelligence algorithms. You can think of them as the leading scientific hypotheses of aspects of the mind. So these are specific in silico neural networks that can actually work. That is, they can produce behavior that may be rivaling human behavior. And they actually explain the empirical data, not just the behavioral data, but the internal neural data on which they're meant to match as well. And I hope you've seen some of that already in the lectures, and I will take you through that a bit more, remind you about that again today. So again, today I'm going to review this for the area of visual intelligence or perception, especially around object recognition, and I'll try to explain why this even matters at all, why this is important that we take this kind of approach. And when I say we, I'm of course meaning many of the folks that are in my group and others. Some of them are shown here. You've already heard from, I think you've heard from Co and Tiago already. And so I'll try not to repeat things that they have already said, but I'll connect to some of the things that you heard from them to reinforce that. So let me sort of pause here before I get into the guts of this and ask if there's any questions about philosophy here of approach. I think I saw a hand up, but maybe, nope. Okay. So you guys are all on board, I guess. So this is, okay, this makes sense here at CBM Summer School. Good, so you're buying the philosophy so far. So, okay, let's go back to visual intelligence. What's in the scene and so forth. So it's known when you start thinking about a problem like this, and when we started working on this about 20 years ago, that you don't actually just absorb that whole scene at once and say, well, all the cars are here and all the people are here. You can't, you have the kind of impression that you're doing that, but when you measure things carefully, you don't actually do that instantaneously. What you do is you have a high acuity sensor in the center of your eyes, your so-called fovea, whereas if you're looking at this spot here, you have high resolution near the center of gaze. And the parts of the brain that most work on things like object recognition, they're very connected to this sort of central part of the retina. So they focus on the, say, central 10 degrees, which is what I'm showing you here. It's like two hands at arm's length is about 10 degrees of your visual field. And those are the set of brain regions that we've been working most on. And the way that you you digest the whole scene is your brain makes an analysis of this central fixation point, but it's also in parallel making analyses of what's out there in the periphery. And by doing that, then it's guiding eye movements to sample other parts of the scene. And you probably heard some of this in Gabrielle's talk yesterday. So you make eye movements to sample other interesting parts of the scene. It's again doing analyses in the central 10 degrees of window, central 10 degrees or so, to then guide future eye movements. And so these eye movements are called saccades. At each dwell point, you dwell for about 200 milliseconds. So again, this is background. The reason that this matters is it allows us to operationalize how we study things as tasks and vision in a way that's aligned with the neurobiology. So specifically, we can study 10-degree images for durations of on the order of 200 milliseconds, and that's very much in line with the natural processing regime of the biological system of interest, ourselves, primates, and their non-human primate cousins that I'll show you in a minute. So here's to help convince you of that. This is removing those 10-degree snapshots and showing them just in a sequence. So now you don't get to make eye movements. If you just stare at the center of this, this is as if someone's making eye movements for you. Okay, hopefully that played for you, and hopefully you were able to notice that in these glimpses, if you will, you were still extracting, oh, that was a car, that was a sign, that was a person, even though you were no longer directing those eye movements yourselves. In other words, your brain is capable of doing that, even if it's not directing eye movements. And that's a actually well-studied phenomena. For instance, you can read faster if someone moves the reading for you than if you actually move your own eyes. So your brain, your visual processing systems that I'll tell you about are able to keep up with this. And the eye movements serve to sort of direct them to other interesting parts of the scene. And they're limited by motor times and so forth. So, okay, so you can then ask now these questions. I've narrowed this down to you. Instead of having a whole image, I can now ask what's going on in just the central 10 degrees and try to get at some of the computational guts of how this works here in these central 10 degrees for 200 milliseconds. Again, asking questions like, is a car here? Is a person here? Is a sign here? So, and so we call this, and you've already heard this, I think, I'm just saying this as a reminder, we call this core recognition because we think it's a core component of visual intelligence, and it aligns with the neurobiology in the way that the system operates and the way that I've sort of outlined for you here today. Okay, but I want to remind you, this is not all of visual intelligence, of course, it's just a core component. Okay, and I've been testing you as primates, Homo sapiens, as you do this. And we, of course, test lots of Homo sapiens as they do that, lots of other humans. And the way we often can do that is tests like this. This is just one example test that we like, where I just ask you to stare at this dot, and I'm going to show you a test image, and then you're going to report among two choices. So I'm going to give you just 100 milliseconds of viewing time, and these are 8-degree images. So, okay, you can try it yourself. Here we go. Hopefully, if you sort of say automatically, go, that was more likely a bird on the right than an elephant. Here, we'll try another one. More likely a face than a car. Notice that face was sort of angled on a crazy background, yet you're still able to do that. Here's another one. Okay, so that's a bird again, natural image. We can do this all day long. And in fact, we can get millions of behavioral trials this way. And as we can quantify your abilities quite well, you can do this for synthetic images where we control all the latent content, the position, the pose, and size, scale of the objects, like in the above, or more photographic images from natural databases shown at the bottom. And those are, of course, things that we do. So we measure all that. And what's most interesting about humans and their ability to do this is you do this in a way that generalizes well. So this is called the invariance problem. And this has been known for some time, that you're able to say this is a car, even in the face of large amounts of variation. So you can still say all of these are cars, or this is a car with the background clutter, or this example here. So all these different types of variation, these are images that you probably, unless you've seen my lecture before, you've probably never actually seen this image before, yet you know this is a car right away. So you don't get the exact same pixel map, yet you still generalize and say there's a car there. And that's the core generalization component of recognition. And that's what made us better than machines, at least in around 2010. So here's human ability. This is performance on the y-axis. And this is as we vary the uncertainty, the amount of generalization needed to solve the task. So on the left is kind of like you're always seeing the car from a perfect side view at a certain size. And on the right, you're seeing cars in unknown poses and sizes like these example images shown at the bottom. So high uncertainty. And you see humans, you have a little bit, you're very high performance here, near 98% accuracy. And you fall off a little bit when you get more of this. But machines, they do quite well in these kind of very aligned situations, but then they fall off even more rapidly than humans when you get to these high uncertainties. So this was the motivating question for recognition. How do you generalize over that as well as machines? And this is when we say core recognition, I don't just mean the central 10 degrees, but I mean the ability to generalize in the way that I'm describing here. So that's the kind of test images that we use. Okay, so again, all motivating kind of background in a way. And I've told you about humans doing this so far. I've been talking about that. And I implied that we're better than computer vision systems at doing this. And in fact, in the days that we started this, we were far better. We are now sort of in a regime where we're not entirely better. In some ways, we're matched on some of these tasks, and it's much harder to start to see the differences between us and computer vision systems, but we think they still exist, and we're happy to talk about that. So I'm still going to leave us as better than computer vision systems on these kind of tasks. Okay, but the rest of the talk, I want to tell you, again, even though we're interested in the mechanism of human vision, I'm going to tell you about this other species, the rhesus monkey, that we use as a model of this. And these are both primate species, of course, on the left, ourselves and our primate cousins. So you might think we share, because of evolution, a lot of similarities and mechanisms. And in fact, there's a lot of evidence to support that. And here I'm going to show you some behavioral evidence to support that we can use this monkey as a model of this. This is, again, a non-human primate in our lab. This is up in the animal colony doing the task that I just asked you to do. This animal has been trained to recognize objects. It takes it a couple days to learn each new object, like a car or a person or a table. And here you see the animal is triggering a touch screen to generate a test image and then making a choice among two objects, just like you were doing there. And every time it's flashing green, this means he's getting it correct. And every time it flashes black, he's wrong. So he's not perfect. In fact, his patterns of errors, as I'll show you in a minute, are very similar to our own. But they'll generate thousands of behavioral trials of this all day long and do this, and they kind of find it and more entertaining than just sitting around their cage here. And they do this for us. Okay, when we look at, I see a question. Go ahead, Tiago. Yeah.
Hello. Yeah. So in the example that you gave, the human performance here, is this also for non-naturalistic images? Like if we were shown objects like blobs or something, I don't know what would a non-naturalistic image be, but.
Yes, that's a great question, and you sort of answered the interesting part of that yourself. Was like, what counts as natural? These data are from 3D models like cars and other objects that you would probably call non-natural. I think you would call this car I think you mean things that we might have seen or not seen before is what you're getting at. So Josh Tenenbaum, my colleague, would say you've never seen a tufa before, and he'd create an object out of a set of things. Now, is that novel? It's sort of related to things that are novel or not. So it's hard. Defining novelty is tricky, but these data here are exactly are drawn from 3D models, which almost most humans would find them as not novel. But again, you may have never seen that particular car model before. So it's hard to fully answer that other than to say what I just did. But I think you're another way to phrase your question is, what if I give you random collections of pixels? Like, forget even objects at all. Like, then humans are actually pretty terrible at this. So there's something that's still about object structure that is important to have here to do this. And so I hope I've answered your question, but please ask again if I didn't.
That helps. Thank you. Okay.
But it's a good point. Like, we are not good at everything better than machines is another way to take it. This is an example of something we're good at. And what you're getting at is how much of that is learned versus built in is sort of implicit in your question, I believe. And maybe we can return to that in discussion. Okay, for now, take it as an observation that we're good at that. And so here we go. And all I'm telling you here is our primate cousins are also quite good at it. In fact, they're good at it in ways. This is the kind of confusion matrix. Red means that you're tending to confuse objects, like, I don't know, hangers or knives and forks might tend to be confused more than knives and bears. And that's sort of intuitively obvious. That's what's on this plot here. The colors is the degree of performance where blue is good and red is bad. And what I want you to see is that the patterns of what we find difficult, the reds, are similar to what the monkeys find difficult. Again, this is just a way of saying it, what we call the object grain. Each of these squares is averaged over many images of knives and forks and so forth. This is at the object grain. Our patterns are very, very well aligned. And again, if you believe in evolution, this shouldn't be that surprising. It's just a kind of quantification that we're almost exactly aligned quantitatively with the rhesus monkey. And this is true even if you look finer at the image grain. Again, this is at an even finer grain, image by image, where do monkeys make mistakes and where do we make mistakes? Okay, so this is just reinforcing the basic idea that you can think of these as equivalent. This model is not just a model in the sense that, you know, a bacteria is sort of a model of our cell. This means it's a model, the systems level model of this visual intelligence ability is intact in this organism. In other words, if we could build a copy of a monkey visual system, we'd have a very good copy of what's likely going on in the human visual system is the way we take this data. It doesn't mean that humans are equal to monkeys. It just means over this domain of visual recognition abilities, we are almost identical and again, probably share the same mechanisms to a very high degree. Okay, and again, both of us better than computer vision systems. So we're going to say primates on the right and computer vision systems that are always evolving on the left. Okay, so why am I doing this? Why do we go to monkeys? You know, I'm interested in humans, you're all interested in humans. Well, it's because we know a lot more about the monkey visual system than we do about the human visual system, and we have much greater access to that system. So it allows us to do much stronger empirical science, and then knowing that what we figure out in the monkey is going to translate very readily to what's in the human. So decades of neuroscience have already given us guidance here of what's going on in this monkey visual system, and I'm going to review that for you. I think you've already seen some of that today, so this is just a very quick overview of that. I don't want to make a redundant talks for you. Okay, so there's the monkey brain. That's the monkey brain on the right. The colored areas are the so-called ventral stream that I'll show you again in a minute. Here it is, ventral visual stream, which has both feed forward and feedback processing. These are, of course, the cortical areas. There's, of course, the retina out in the eyeball that's not shown, the LGN in the center, and then you get to these cortical areas. And I believe you got some of the retinal physiology from Tiago earlier. So the parts of the brain that initially were interested in is these high levels of the ventral stream called infratemporal cortex. And the reason we were interested is that lesions here produce deficits in things like visual recognition. But I kind of want to back up to say, how do neuroscientists even know about these drawings that I make for you here? And I'm just going to kind of give you a bit of a sort of whirlwind overview that things that you should know. That's a rhesus monkey on the left. This is the rhesus monkey brain when extracted. And you can take this brain, and now it looks like just a mass of tissue. You slice through it and you stain it. You can start to see layers here, right? So you start to see these different layers. You can almost make out individual neurons with little tiny dots in this image. You zoom in, now you start to see the individual neurons, little dots in here. Notice they're organized into different layers. Okay, these are called cortical areas. Each cortical area has a layer, that has multiple layers, 6 layers in it. Just for those of you who know about deep networks, of course, sometimes deep networks have their so-called layers. Those in the models correspond to areas in the neuroscience language. So that's a bit of confusion in the field, but just so those, if you don't know, the visual cortex has different lamina in it. And most of the deep network models that you've heard about that approximate the biological systems have not yet modeled in detail the laminar processing going on within each cortical area. They've only roughly approximated that. But just to keep going, you can zoom in and say, if you stain a few of the cells, you can see the cells are not just cell bodies where they're stained as dots, but they have very long, dendritic processes that are thought to do complex processing. Some people think of neurons as full integrated circuits themselves, not just point process integrators. That's one idea in the field. But what I'm bringing also is to say we can go in and put electrodes and drop them in the monkey cortex here and record the spikes of individual neurons that are sampled, say, in a cortical area. So randomly sampled neurons like this one visualized here. So there's a neuron with its dendritic processes. Now let me pause here to ask the audience, like, why, I'm recording spikes. We could record, calcium levels in the cells or gene product levels of various types or many other things we could measure in the biology here. So I'm showing you a bit of anatomy, and then I'm saying we're going to make functional measurements of neurophysiology. It's called the spike measurements out of neurons. Why does that, I want to ask anybody in the audience, why is that a useful thing? Why does that have privilege over some of those other things? Or does it have privilege? Because I'm going to talk about that next. So it's important that we think about that. Anybody want to offer? Why, if you could only record one thing, would you record spikes or would you record gene product levels? And why that? Why would you make that choice?
I mean, I'm just thinking of different kinds of brain measurement, like, techniques. And is it just because of the temporal resolution you get from spiking activity?
Well, yeah, let's keep going. So what do you mean say the temporalization? Why is that important?
Maybe there's different, when you're trying to recognize an object, right? Maybe it's important to get, depending on like what kind of object it is, you want to be able to measure the activity at different timescales to be able to see if there's, if certain objects require, neural activity in like a smaller time window versus other things that might be kind of, you might want to just average over like larger time windows. I hope that makes sense.
Yeah, it sort of does. The time is a key thing, right? So I think that's a key point. But there's something that's like, remember, if we're studying, I didn't say this right, we're studying visual processing, like visual recognition, which I told you takes on the order of 100 milliseconds, right? So things like gene products, even calcium levels, they don't vary that fast, right? So if you're thinking about the mechanisms that actually enable that behavior, they got to be fast. They have to be in that time scale. So this is the only known, these spike processes, these transmissions of spikes between different neurons are the only known ways that neurons are able to even transmit, what's on the retina, all the way up to actually to execute on behavior. So they are the ongoing processes to enable the behavior that I set up. It's the only thing that's sort of fast enough to do that. So it gives them privileges, even though the brain has other things, and if it learns over days and weeks, all those other things become important. But when you're studying visual processing, the spikes have a privileged are privileged in that regard, and that's the only units that can transmit the information at the rates that are necessary to explain the behavior. This is important if you're interested in things like learning or other things. You might think about all those other things matter too as the system changes because it's much longer timescales. But as the questioner answered, the time scale is really important here, and that gives spikes some privilege. So it's not just that they're easiest to record, they are one of the easier things to record, and it's an old technique, electrophysiology, but it's also maybe fortuitous that that's actually one of the most relevant things against processing like visual processing. I hope that makes sense. And if there's maybe further questions, just put your hand up here. I see one person nodding. Everybody okay? Okay. So I'm just setting that context here, but I'm coming back to anatomy. So monkeys, this is a monkey visual brain, a visual system. Why do we, you've probably seen drawings of these different areas. I've been showing you some of them. They're on the left. These are different topographic areas. You've probably heard about visual area V1, the first area, and V2. But kind of how do we know that, V2 is like downstream of V1? I just want to remind you, because I don't think this has been covered, the reason we know this is because of decades of neuroanatomy where people make injections in visual area V1s here in the back, and then they look at where those projections go in areas like V2. They see certain patterns of projection. So this is showing the pattern of projection from V1 to V2, and you see especially these white areas where you have dense innervation from V1 axons intervening neurons in layer cortical layer 4 of V2. So that's a so-called forward projection pattern, like you see here schematically. Upper layer, layer 2, 3 neurons projecting to sort of center layer 4 layers in V2. And if you do the flip experiment where you look what goes from V2 to V1, you don't see a symmetry here. You see this sort of pattern where the upper level neurons, for instance, project to the upper and lower levels of V1. So I hope you see this asymmetry here. And this has been called a forward pattern because V1 gets most of the dominant input from the LGN in the retina. And so then this is a continuation of that, and that's called feed forward. And this is called a feedback pattern. And why am I telling you all this? Because it's been known that you can do this not just for V1 and V2 and establish this pattern, but people have done all the different visual areas. And here's a whole bunch of them listed here. And you can establish a so-called hierarchy based on just these kind of neuroanatomical studies. Functional studies also back this up, things like latency as you measure neurons in different areas. But the neuroanatomical studies essentially made the foundation of this kind of hierarchy. And this is now this is a review from ours about 10 years ago showing the ventral visual stream. Here is that hierarchy again, V1, just showing the feed forward flow, V1, V2, V4, and these so-called infratemporal areas that I mentioned here in yellow. There are other visual areas here. These are dorsal stream areas on the left. And what I've colored is sort of the parts of the brain involved in the central 10 degrees of processing. That's what the colors mean. Again, you can see even V1, most of your V1 is devoted to the central 10 degrees. It's sort of surprising, but this is what, kind of the density of cells that you have in the center of your retina projected on the cortex. And so it's something important to keep in mind. I'm not showing the feedback patterns here. I'm showing you the latencies, approximate latencies. As I said, those are the functional latencies of the spikes that are consistent with this kind of anatomical patterns that I showed as well. So again, this is a review of the sort of neuroanatomy of the ventral visual stream with a bit of neurophysiology layered on top. Okay. I think you've already seen these kind of things and heard it, but I just kind of wanted to remind you where it comes from. We'll talk a lot about the infratemporal cortex here. You've probably already heard this, but there it is at the top, a collection of these three areas here. So back to this view, this is the ventral stream. Zooming back out, this is the ventral visual stream, infratemporal cortex, a collection of roughly 3 different regions. And just to orient you, the brain doesn't just end there. Of course, these neurons project areas involved in the frontal cortex, which are involved in decision and action, so maybe executing a choice movement, and to regions involved in the mediotemporal lobe, which are thought to be involved in value judgment and long-term memory formation. You remember Teuber said where emotion and memory enter, right? So there's sort of hints of that here, right? So these this is just to give you the lay of the land of the ventral stream. And now I'm going to kind of take you through some conceptually how we think about this, given that anatomical background. Here are the kind of millions of neurons schematically laid out here in these different areas, just laid out here where there's both a feed forward and a feedback flow. I think you've seen this slide before. There's of course recurrent processing within each different area. And the way we conceptualize this is when an image strikes your retina, it's transmitted, as you heard in earlier lectures, those pixels are converted one-to-one to a map on the back of your retina and the retinal ganglion cell firing rates. So photons are converted to spike rates, which are schematized here. You can think of these as the high spike rate neurons in the retinal ganglion cells and some low spike rate neurons as schematically illustrated. So a population pattern of neural activity. And that pattern is rapidly transmitted and reconfigured through the ventral stream into a new pattern of activity. Here's a new pattern up in IT cortex that takes about 100 milliseconds to go from when the image strikes the eyes to when these neural patterns are activated. Here's a specific neural pattern in IT. Again, red might be high firing neurons and white would be low firing neurons, just as a schematic here to give you a sense. And that takes about 100 milliseconds. Again, that's in line with the time scale that I showed you earlier of the biology of when your system, when you are exploring the scene, you dwell for a couple hundred milliseconds. So your system is able to complete processing of that region, at least through the ventral stream, easily within that time scale. Okay, so then we can go to a new image and it produces a new pattern. And if we go back to so it's a new pattern, it's a different pattern. It's not a copy, it's not a photograph of this, because that's what's on the retina. It's some transformed nonlinear representation of this original image. And if we go back to the original image, it essentially reproduces the pattern that I showed earlier. So it's not as if these are just random patterns being spit out of the ventral stream. They are just nonlinear transformed patterns of what's striking the retina. And we can go back to this image here, which of course we see as a giraffe, which is, again, going to reproduce the kind of giraffe-like pattern, if you will, in IT cortex. Again, not a photograph of a giraffe, just a pattern of neural activity. Okay, and we call these patterns representations. You probably hear that word. That's what a neurophysiologist means by that, is the population representation. It's the pattern spikes across a set of neurons, like the IT representation is the pattern in IT. And here's a kind of RSVP movie that's going along at, again, faster than 100, it's about 150 milliseconds per frame. And your system can easily keep up with this IT. This has been well shown. The ventral stream can easily follow at these kind of presentation rates, which is very much in line with what I told you at the beginning about your ability to do this quite quickly, as I showed you earlier. Okay, so this is, again, by way of context, I've been sort of emphasizing a kind of feed forward view of the system here as I've been talking, and we'll return to that at the end of the talk. And you heard some of that in Kohei Ichikawa's talk about the recurrent processes as well. But this is the feed forward kind of baseline view of the ventral stream. And the way we know all this, by the way, and I maybe should have said this earlier, is that monkeys, we can test them. They stare at a screen like this one, and we show images to them. Maybe they're doing a task, maybe they're just staring at it. It actually doesn't matter that much for the ventral stream. You produce these patterns in IT cortex regardless. If they're just staring at the screen and they're awake, these patterns will come out in their IT cortex and we can measure them. And I'll show you how we do that in a minute. Okay, so you go and measure the spikes with electrodes that we discussed, and we discussed why that's important. Here's an example recording in IT cortex, recording from one neuron in IT cortex. Here's some spikes. Each dot is a spike in response to this particular image. If you repeat this image over and over, you see, as I said, you get essentially this is just one element of that representation, one neural site. You get essentially the same spike rate. Now, notice I say essentially, it's not identical. There's certainly some variation in these spikes, but you see high rate with a latency of about 100 milliseconds in response to this one image. Here's another image, again, generates high rate at about 100 millisecond latency. Here's a time scale bar. The image is on during this white bar here, latency of about 100 milliseconds. Here's another image, a little less rate, a little lower, and then here's one that's a little bit lower. If you go to another neural site, you get here's a different pattern. It's not as if every IT neuron does the same thing. That would be boring. Here, it doesn't like this first image, responds higher to this second image, and medium to this third, and medium to this fourth. And here's another one which it really likes the fourth image for some reason, and less so the first two. Okay, and so these are the kind of data that you can record out of IT cortex. You can record similar data from any level of the ventral stream. I'm just showing you IT. And what I'm going to do for a lot of the rest of the talk is just talk about these as if they're just the mean rate that's coming out of each of these things. So this is site 1, and I'm going to just sort of average all the spikes in here, averaging over about 100 milliseconds of time, and over all repetitions that we've collected here. Here's 10 trials. And that allows you to say the mean rate of site 1 in response to image 1 is, say, 60 spikes per second. So it converts all these fine-grained spiking data to one number. Now, there are a lot of assumptions in why we should do that. And those are interesting assumptions that we should unpack. But it turns out that this is a good way to start on getting traction on the system, because these mean rates turned out to be pretty good at predicting the kind of responses, the kind of behavior that you're going to get out of the animal in terms of its mean ability to do things like recognition. I think you've already heard that from Ko, and I'll review that for you in a minute. But here I'm making a change that we can, I'm making an assumption that we can relax later, which is just average over this time scale here. This converts all this complicated spike data into, again, in this case, 4 numbers out of site 1, 4 mean rates. Okay, and those mean rates are what I've been sort of implying for you here. Is think of these red as sort of the level of the rate of each of those neurons in response to each image. All right, and we do this is IT cortex, and we do this not just for single sites like I've been showing you, but we typically in our group, we put in multiple arrays where we can record hundreds of sites simultaneously. Remember, there are millions of neurons in IT, so we can't record them all. We take samples that are about hundreds, or we can make samples of about thousands when we record over many animals. These are put in, this is the animal's IT cortex in a sterile surgery. Here's the skull, here's the dura mater, which has to be opened up. These arrays get implanted, the dura gets closed, a protection puts over the skull, muscle put back on top, and then the animal has a connector on its head that we can just record from the same locations for many months in this particular prep here. And these are so-called chronic recording arrays. Okay, when you do this kind of experimental technique in a monkey, then you can now get, again, hundreds to thousands of sites. And here I'm just showing you the mean rates from over now. These are neural sites in response to one image. So these are real data that are meant to be kind of aligned with the schematic I was showing you earlier on the left. This is the sort of population pattern, the representation, if you will, of this image over a population of neurons in IT cortex. Some responding high in green color, some responding low in black, which corresponds to the red and white on this left side of the plot. Okay, that's called a response vector in our language, and I hope you've seen this before. I think you have from others, just reinforcing here. Here's not just one image, but here's 8 test images. So these are just arbitrary images. Notice that you don't get the same pattern out each time, which is, again, in line with the schematic I've been telling you about. These are real data, again, on the right. But what's cool now is we can do not just 8 images, but we can do thousands of images. And now I'm not even showing you that, can't show you all those thousands of images here. Here, they're grouped by the category they happen to be in. That's what these white lines are about. And you see some structure at the category level, but even just with your eye here. But this is really just to orient you to kind of fundamental data that we sort of start with on a lot of the analysis that we've done that I'll show you next. I'm going to pause here just because if there's technique questions about how we get to this point, this is probably a good time to take them.
There's someone online that would like that you discuss a little bit more about the role of feedback, but I guess maybe you're going to address it later.
Right. So that will co-discuss that a bit, I hope, in an earlier lecture, and I have a bit of that at the end. So again, this is a review of the ventral stream. So let's just say I haven't said anything about. I've kind of given you the feed forward and the feed. I've told you this feedback in the system. I've described the latencies of it, and here's the neurons that you get out. First of all, these neurons, we don't want to say these responses are entirely the result of feed-forward processing. These are just the observations that come out of IT cortex. In fact, we know from Ko's work and others that these neurons are modulated by recurrent processing as well. So this is sort of a kind of an average of both feed forward and recurrent / feedback processes in creating this observable here, which are just the empirical spike rates coming out of IT cortex. So this drawing here alone is not a statement about feedback, it's just a telling you of the kind of empirical data that we collect. But as I go forward, if I start to make claims that you think lean higher on feed forward versus feedback processing, then I hope someone will bring that question up again. For now, I just want us all to be aligned on kind of what kind of data come out of these systems. And this is all I'm trying to show you here. Okay, now let's go to sort of like, why is this even important at all? Okay, you can stick stuff in monkey brains, you measure some neurons, you talk about them like patterns of firing out of IT cortex. It's built on a whole bunch of neuroanatomical history. You told me a monkey and a human are similar. Why does this matter? Well, the reason this matters is that it's been shown, and we and others have shown this, and some of the references are at the top here, that if you just build linear decoders, which learn on a few training examples, on the orders of tens or maybe hundreds of training examples, that testing on new images, new test images, those linear decoders, if you measure the IT patterns for those new test images, they predict the patterns of behavior that you're going to get out of the organism, whether out of the monkey at least. And again, we think that's analogous to what's going on in the human. And so that what that means is we think we have an approximation with these linear decoders of how the neural activity in this part of the brain ultimately is driving the behavioral choice of the animal and these recognition paths. Another way to put that is the neural population representation explains and predicts the object recognition behavior in the sense of you first apply linear decoder, which is a population readout, a simple population readout. You have a set of decoders, one for each category. That's typically how this is done. And then you test on new images and you ask, how good is that decoder? It turns out to be quite good, but no better than the animal. So it's good and bad in the ways it should be good and bad if it's aligned with what the mechanisms that are going on in the monkey or the human brain between IT and the behavior itself. And there's a lot of assumptions when we do that, right? We're taking mean rates. I've already told you, we were assuming it's out of IT. In fact, if you do this same procedure out of areas like V4, V2, you don't match the behavioral patterns. So, and Ko had told you some of that were ongoing work. This is not a final statement on the mechanisms of how IT gives rise to patterns, but it was really quite motivating that already at this, you could explain a lot by just linear readout of IT to explain the animal's patterns of behavior. And so that the parameters of that actually are really interesting as we work them out. And again, Ko and others are working on that because if you think about that, those linear decoders going between IT patterns and the animal's behavior, they're essentially a statement about how neural activity inside the animal's head is giving rise to its reported percept. And that has predictions in it. If you were to perturb that IT activity in certain ways, you would cause the animal's percept to change in particular ways that the linear decoder models tell us. So, and now it gets into things like brain-machine interfaces. If I was to inject current, activate neurons, whether electrically or optogenetically in IT cortex, I should be able to inject certain object percepts. If you think about the movie The Matrix, if you guys remember that, the idea that you could plug into the brain and create a virtual reality. These are the kind of models that allow you to begin to explore those ideas, so-called write-in ideas that have been around for a while. Now these decoders allow us to begin to test some of those. I'll just say in passing that the hard part about those experiments is not necessarily the models at this point. It's just actually controlling the neurons at a fine scale and doing that in a reliable way. And there are many labs starting to work on that now. But just for context, that's one way to think about why that matters at all. And I just said that here. Okay, but if we come back to intelligence and the thing I said at the beginning of how the visual system extracts identity from the world, let's call that the AI relevance of why this matters at all. It says that, well, whatever that primate patterns that I showed you at the beginning, where they're better than machines, it's somehow because they're able to produce these IT patterns. Their IT patterns are kind of the neural basis, if you will, of their good generalization performance. In other words, if you had a machine that could produce a representation of images that looked like the IT representation, it would be as good and no better than the monkey or the human at doing those tasks. And so if you're an AI person, you're like, well, if I could just figure out how to build a nonlinear processor from the image to IT, then I basically have something that works like a human or a monkey. So that's what's sort of motivating on the so-called encoding side of that from the image to the IT neurons themselves. So, and again, I sort of already said this, but a key takeaway is that mean firing rate assumption of IT, because it matches behavior, it sort of reinforces that was probably a reasonably good starting assumption to make. The one I said, we just average over some time window. And I say reasonably good. It's not perfect. In fact, that's active area of research in the lab. And I think Co told you some of that. Okay, so again, the motivating question from this is once you accept that the neurons explain the behavior of interest, then the motivating question becomes, well, we all know what linear decoders are. Those make sense to us. We can imagine where they live in neurons. There are questions there to still explore. But the interesting computational questions are, how is this IT representation, as I said, computed from the image? So there's some nonlinear computation from the image to IT. And similarly, what are the intervening representations, if you will? This is another way neuroscientists would frame this. What's happening in each of these levels? Those are the same question, just said different ways. Okay, and there was, Tiago has his hand up, so we'll let go, but I'll just sort of say, there was a bit of a breakthrough in 2013 on this front that you've already heard about that I'll review for you briefly. Tiago, go ahead.
So, hi, is it known whether these representations or how stable these representations are across sessions or days? If you
yes, so when you say these, you mean the IT patterns that I'm showing at the top. Yes, and it's nothing in biology is fully known, but what people have, and David Leopold's group is probably has some of the best data on that, where they implanted IT neurons with chronic recording arrays for many months, recording from the same sites, and showed remarkable stability of these patterns over many, many months. Our group sees similar things where we can record at least for many weeks and sometimes months with very similar patterns. What makes it tricky is that if you see a change in a pattern, it's hard to separate movement of your chronic electrodes from some actual change in the pattern. And so the picture you have in our head is that it's very stable, but you should not think it's 100% rock solid stable, because we've also shown that under certain manipulations, you can bend these patterns. So I think the answer is kind of like all things in biology. It's mostly stable, but not entirely stable, but it's certainly stable within a session. Again, I sort of wanted to emphasize that you shouldn't think of this as changing very rapidly. It can change over hours or perhaps over days if you put it under the right conditions, but it's stable, let's say, within minutes, and you keep coming back to the same images again. I hope that answers your question. Sort of it's shades of gray. Does that help?
Yeah, thank you. I had a related question.
Yes.
This time. So if to build a brain-machine interface, the question is the timing of the spikes important, or is it just the mean firing rate that is important?
That is another great question. And I sort of emphasize that the mean rate is kind of what we think is most important. And a piece of evidence for that you're alluding to that I didn't highlight is that folks like Arash Afraz, who's at the NIH, have done ESTIM experiments where they electrically stimulate neurons to try to cause perceptual changes. They've done that in like groups of face selective neurons to try to shift the monkey's percept to report more of a face. And what they're doing is they're just injecting 200 hertz ESTIM. So they're just activating the neurons in a rapid, they're not trying to get the millisecond by millisecond timing, they're just trying to turn up the firing rate of the neurons with that. And when they do that, they cause perceptual shifts in the direction of the animals do report more face-like activity. So mean rate shifts in the activity are enough to induce perceptual changes in the predicted direction. That doesn't fully settle the question. It says mean rates are a big part of it, but it doesn't mean that maybe if we got the precise spike pattern, it would actually make a cleaner percept, which is kind of where your question is lurking. And we don't know the answer to that yet. So it's sort of, again, sorry, all my questions are like shades of gray. Mean rates are important, but they're probably not the whole story. But at what level the rates matter, our guess is sort of on the 10 millisecond scale is probably the most relevant time scale to get things matched. 100 milliseconds is probably a little too broad. And Co's data, again, speak partly to that. I hope that helps answer your question, but that's really not fully known yet. I gave you a bit of evidence of how we think about that.
Thank you.
Let me just sort of get to a break here in a minute, because I think we're getting towards a break, but let me just sort of set up. I think I'm going to go quickly here because you guys have already seen this. I saw yesterday. Basically, we didn't know, even 2013 or so, we didn't have models that could actually explain the neural activity quite well at all. And, we could record these neurons like I showed you, and we could look at them, and people have done this for decades, where you kind of look at, this is the mean spike rate of the neuron, and here's a bunch of images. And this is the, it likes this image of this chair pretty well, and that image of this chair, and it doesn't like this image of a chair less. I'm not showing you that image. It's some image of the chair it doesn't like, even though it has a chair in it. So you might be tempted to call this a chair neuron because it likes chairs more than cars, but you see its patterns of response were not category selected. There's something more complicated going on here. And this is very hard for us humans to sort of, if it's not category selected, we're like, well, I don't know what it is. It's the edge of the chair. There are decades of work of people trying to use words to describe what makes these neurons go up and down. And that was kind of a dead end. It didn't go much further than this, and we just couldn't explain things very well. There are so-called face neurons, same thing. Yes, these neurons like faces, but not all images of faces, which you see here. So it's much more subtle than saying the neurons like faces. It's a good starting point, but it doesn't explain the neural patterns quite well. And then here's area V4, that one of the intermediaries, even more complicated, even less categorical. You wouldn't want to say chairs, cars, or planes or anything. It likes this chair and that car and this image of fruit. So it's probably some intermediate features that people were guessing at. And again, you saw some of that background yesterday. But the core thing I want to tell you is, like, if we don't have models that can actually explain and predict these, that is, you put up a new image and you're able to predict what the neuron's going to respond to, then we can't claim that we have an understanding of what's going on. So that was a motivating question for us, is to build models that could capture these kinds of data and predict them for new images, to build new encoding models. And this was not just a question of us, this was motivated by the field for many decades. This has been the goal of visual neurophysiology, again, for decades. And most of the work has been done in early visual areas like visual area V1. You've probably heard from Tiago, where people build these kind of Gabor-like models that are tiled across the visual field. These, because they're tiled across the visual field and repeated spatially, these are, can be thought of as convolutional operators. And current models of the system are convolutional, and this is motivated by early physiology. Different orientations at different positions. All orientations represented at every point in V1 was the idea. There was known ideas of, you have a linear operator with some nonlinearities, rectifying nonlinearities, and normalized gain control. These are kind of known things. Again, here's a review in 94. And these are all approximations at the level of single neurons at just kind of the early levels of the visual system. It was known, as I've outlined, that there's a deep stack of areas, and there's probably a similar style, if you will, of linear and nonlinear operators reflected across the ventral stream. It was thought that feed-forward was a big core component of what was going on based on the latency and some of the data that I briefly showed you, although the recurrence was known to also be present. It was thought that rate codes across the population were the way the information is contained, not individual neurons, but the combinations of neurons. These were things that neuroscientists knew and kind of part of the dogma of the field of how the visual processing worked. So decades of work kind of would say these kind of things. And this was motivating many kinds of models. One of the earliest ones is Fukushima's model in the 1980s. I think you've seen this already, of an artificial neural network that's trying to approximate areas like the ventral stream. This was followed up by some really cool models from my colleague Tommy Poggio and his collaborators, this HMAX family of models that try to take this further than Fukushima had done. And there was some good progress there on building invariant properties from these kind of models. And then some folks in my lab were started to try to build even more advanced versions of these kind of feed-forward mostly models, these sort of deep convolutional models here. This is 2008, 2010. And I want to stress here, all this model building is important work. Many of you are building models probably right now or thinking about that for your project. This is some of the most important work in basic science because these are the hypotheses of our field. Even if they're wrong, putting forward testable hypotheses is a core component of scientific progress here. And these are all examples of mechanistic hypotheses in my mind of how the system might work. Unfortunately, the problem was these models from 1980, 2010 or so, some of which were in our lab, and I'm not afraid to say, they were all not good enough. They were all inadequate in terms of predicting the detailed patterns of neural responses. We're well motivated and fundamentally have a lot of truths in them, but they just were inadequate at a quantitative level in explaining the neural activity patterns that I motivated you with here. And that's what I just wrote here. Let me show you that quantitatively. Here's explained variance of a cortical area V4. Here's a bunch of those models that I just outlined for you at the top. And you see this is 100% explained variance at the top. If you were perfect, you'd be all the way up there. And you see the models are down here in this 20% range or so. So here you can see the failure. Here's the prediction of one particular model in red. Here's the actual response of the neuron that the model's trying to model in black. And you see the red and the black don't align very well. And then we quantify that as explainable variance, which is on the y-axis here. This is, again, one model. This is one neuron out of one model trying to fit one neuron in V4 as just an example. I want to point out, explain to us means predicted. This isn't fit. This is predicted on new images, and that's important to saying you have a model in a real sense. It generalized to new images. And this also accounts for noise levels in the data, by the way. Okay, then if you go, Tiago's got his hands up. Go ahead, Tiago.
Just had a question. And that is about how much variance would we expect one person's neural firing here to explain in another person's brain?
Yes, so this is a great question. We call this the Many Monkeys Project, or the so-called ceiling project, which is kind of like how far, even though I'm saying this 100% number is like, I would like a model to explain this. The question is essentially like, put it this way to us, is like, should an IT neuron should my head be able to predict what's exactly in your head at the level of IT cortex? And that in some sense is the level of modeling that we're actually after. It's called a species level model. It's like, I shouldn't expect a model of a system that's trying to be a species level model to predict the individual variations. It's just trying to predict across from one organism to another. In other words, to put it as a model is trying to masquerade as a member of the species, and want to check it by that kind of standard. I think that's the sort of notion of your question of follow, right? And Ko has actually done the leading work on that, because he's there in the audience with you, and he can say more. He has like 6 animals recorded, and he's done things where he's asking how one animal predicts the other animal. And I don't want to give you that whole story now, other than to say we know that none of the current, certainly none of these older models are giving you here, but even the newer models, none of them are yet even at that ceiling level that you described. That being said, that ceiling level is a little bit lower than the one I'm showing on this plot, for the reasons you can imagine, that there is species level variability there. So I'm not ready to give you that lecture, but I think it is a good motivating question. And I'm just trying to frame it so everybody in the audience is sort of following along of like what the goal of that means. And I can't read the audience here to say if I kind of fully answered it, because this is a complicated topic. But I'm happy to stay on that longer if I kind of left you guys more confused than explain.
Can I just comment something that maybe that's something also that would depend on the area, right? Some areas we would expect single neurons to better or more fully predict neurons in another subject, while as maybe we go higher in the hierarchy, that may not be the case, right?
Yeah, that's the usual intuition. In fact, that's kind of, if you go to areas like hippocampus and areas where everybody's memories become much more unique, that you'd expect more idiosyncrasy as you go higher, right? So beyond IT is perirhinal cortex, then hippocampus or entorhinal cortex. So we're sort of at a high level, but we're still in visual areas where there still is cross-species predictivity. And the way you do that, and Ko could speak to that, is you can take an individual neuron out of one monkey, and you can take a bunch of neurons out of another monkey. You can ask, can I make a linear map from the so-called source monkey onto the target neuron in the new monkey? And those linear maps, then the ability to make that linear map is a sort of measure that you're after. How good are those, as that explained prediction? And that's the way we set those baselines, is by making those kind of predictions. That's why we call it many monkeys. One monkey to another monkey, and that sets a ceiling. If we were going to take a model to a monkey, it should be no better than that ceiling. And as Tiago points out, that number is probably higher in the lower areas than in the higher areas, but having full quantification of that is actually not something that's been fully done yet. But again, Ko, I think, is one of the leaders right now in trying to do that, at least in areas like IT and beyond. So you should ask him this offline, if you will. So let me just say, though, that this failure that I was on the slide is not because of idiosyncrasy. Like we know that, right? So that it's certainly some of the gap is that, but we think models should still be able to do better than they did on this slide here. In fact, one reason we know that's true is because we've been able to build models that do better than this. So this was by way of background and telling you that in the past, people tried to build models and they weren't able to get this here. And there was a collaborative breakthrough in 2013 that you've already heard about that I'll review for you.